Digestion and absorption in the small intestine. The small intestine is arguably the most complex organ in the digestive tract. That is because it has so many tasks it is responsible for. To start, imagine that the mucus-protected stomach just did peristalsis and squeezed its acidic contents into the middle of this small intestine. That makes the small intestine's first priority not to get burnt by acid. Yet the intestine doesn't have the mucus coating that the stomach does. So how does it accomplish this task? It gets by with a little help from its friends, the liver and the gallbladder. The liver synthesizes a substance called bile, which it then secretes out of a duct into the gallbladder for storage. When needed, the gallbladder contracts and squeezes that bile down through this duct into an entryway in the small intestine, where it neutralizes the acid. What does it mean to neutralize? Acids are substances found very low on a scale called the pH scale. This measures the number of hydrogen protons in solution. In substances where there's lots of H+ floating around, those substances are very acidic. And you can see that stomach acid, with a pH of between 1 and 2, has a lot of these H+ ions in them. And those are what can burn the lining of the small intestine, unless this acid is mixed with its opposite, a base. And bile has the same base in it that baking soda does. So when the bile mixes with the acid, it actually brings the acid back up towards a neutral pH of 7 that won't be harmful for the small intestine. For more information on acids and bases, go back to my original video under enzymes that talks about that topic. Okay, so now thanks to the liver and the gallbladder, the acid has been neutralized. But that still leaves many of the macromolecules to be broken down. Now the liver and the gallbladder are joined by the pancreas, which secretes many digestive enzymes onto the food to break it down. To break down polysaccharides, it secretes a polysaccharidase enzyme called pancreatic amylase. Just like its cousin salivary amylase, pancreatic amylase breaks polysaccharides into 2-part carbohydrates called disaccharides. Then these disaccharides are broken down further by a logically named class of enzymes called disaccaridases, which produces single monosaccharides. To break down nucleic acids like DNA, they make a class of enzymes called nucleases, which break the bonds connecting individual nucleotides. Lipids, like this fat molecule, are broken by lipases into their respective subunits, glycerol and fatty acids. Then, last but not least, proteins, or the polypeptides inside them, are broken apart by proteases into amino acids. So you should picture in the small intestine many enzymes such as this disaccharidase with their perfectly shaped active sites that can break apart these macromolecules down to their monomers, like these 2 monosaccharides. But what can the intestines then do with all of these monomers floating in a big monomer soup? Monomers are critical for our bodies, of course, because to build our own macromolecules, we need those raw building ingredients. This brings us to the second great function of the small intestine, absorption. The walls of the small intestine are lined with tiny vessels. You can see arteries coming into the walls and then branching off into the tiniest of vessels called capillaries, and then branching back and running back to the rest of the body through veins. Monomers from the broken down food in the middle of the intestine can travel across those walls into the blood vessels so that they can get carried to the other parts of the body. And the form of that small intestine really follows its function, as shown in this photograph of the actual inside of an intestine. The inner surface of intestines are coated with small finger-like projections called villi. Each of these villi provides a lot of surface area for the intestines to absorb monomers. So each of these finger-like projections, zoomed in right here, is a villus, plural villi. And on top of that are these tiny little waves called microvilli. Because of this, if you were to take your intestine and try to flatten it out, the surface, the inner surface of it, would be able to cover an entire basketball court. That means plenty of surface area for the monomers to be transported into the bloodstream. That transport can usually happen either by simple diffusion of small hydrophobic molecules just passing across the membrane from high concentration in the intestine to low concentration in the blood, or for the large hydrophilic molecules, they might need some help. So there would be protein channels inside this membrane that would create pathways for those large hydrophilic molecules to also enter the blood. Imagining these proteins in the walls of the intestine can help you understand how surface area is so necessary. If you imagine a pile of monosaccharides that just got digested sitting in the middle of an intestine. First, let's picture an intestine that just has flat walls. These flat walls could let in some of these glucose molecules if they had protein channels. Okay, now that I drew the protein channels, you can see how those glucose molecules would be able to get to the blood vessels beyond the intestine by just flowing right through each channel. But compare that to an intestine with villi. This could fit many more protein channels, providing many more pathways to the bloodstream. So in this way, an intestine with multiple villi is much more efficient because it can absorb more food more quickly thanks to all that room for extra protein channels on its increased surface area. In this system, the monosaccharides, amino acids, fatty acids, glycerol, and nucleotides will have an easy time diffusing into the bloodstream.